that's happening today at noon here in this room about the Brain Explorers event that's going to be in March. So today is the organizational meeting. Mallory is one of the co-conveners of this plan. So if you have any questions, you would approach either Mallory or me. Brain Explorers is Earlham's contribution to a national Brain Awareness Week, even though our event doesn't happen during Brain Awareness Week because Brain Awareness Week happens during our spring break. It's approximately, it's in the same month. And so this is an outreach event to non-scientists, to members of the community, to kids, to their parents, to students who have never thought about neuroscience, and exploring, getting them to think about the brain. So it's, we hope that each of these stations at Brain Explorers will be some sort of activity, memory, optical illusions, pain, those are the sort of things that have been done in the past. So there will be an organizational meeting to try to brainstorm some of the ideas that could be at the Brain Explorers event, and then to divide into small groups of students who would then coordinate, work with each other to put together an exploration station at Brain Explorers Week. I've mentioned that participation and, that is to say, either participating as a presenter at Brain Awareness Week or participating as a member of the community going to simply sample all of the, or many of the exploration stations at the Brain Explorers Week will earn extra credit in this course. I haven't decided the value, but it'll be points.
Will you give more value to people who present than to people?
No, I don't think I will. I think, well, it depends. I think I would try to make it equivalent. That participation is, full participation is as full and a value as organizing an event. I might be persuaded otherwise, depending on how much effort goes into actually building one of the exploration stations. So there could be extra credit if someone really invests in an exploration station that is a wow thing. Other questions? Okay, then, so that's today at noon. Again, Mallory or Morgan Bailey or Abe Chaudhary, I'm sure I'm not saying that right, are the co-conveners. Let's talk about the exam. So some of the median, the mean and the median were around 80, plus or minus a couple of fractions. So that's pretty good, I think. It was a hard exam. I will acknowledge that it was challenging. And I didn't grade it particularly hard or easy. I graded it what I thought was fair. So if there was a 2-point question and half of the information was there, I gave it 1 point. If it was a 4-point question and I thought it was missing some stuff, I took away a point. And then I tried to ask myself, okay, that's 3 out of 4, that's 75%, that's a C. Is what I'm seeing here really a C level, or is it better than that? And then I would try to adjust by half a point. I didn't go to less than a half a point. I'm just not prepared to granulate any further than a half a point. So I tried to grade it fairly, but not particularly gently. And this is the result, right? So some of you, some of you did really well. And you should, whatever you're doing, keep it up. It's working for you, and do it, continue, please. Some of you are, either didn't invest the time that you needed to do, or missed enough of the material and didn't seek it out from resources available to you, but obviously room for improvement. And I would like to offer myself and academic enrichment and any resources that you know exist here at Earlham to help you improve on the next assessments. We've got more quizzes, we've got more problem sets, and we've got more exams. So obviously there's lots of time for improvement. So from my perspective as a teacher, I thought this was a pretty good examination. It discriminated, that is to say, it evaluated students who understood the material, and it woke up some of the students who were maybe not investing enough time in it. I do acknowledge that it was challenging, but that's a good thing for a 300 level biology class at Earlham College. So there is one student who has not yet taken it because of a family emergency just before mini break. So I'm prepared to hand it back to you at the end of class today, because that student will leave. And but I ask you, if you talk about it with other students, make sure you're talking about it with a student who has taken it. Okay? So I'll hand back both quiz 2 and exam 1 at the end of class today. Okay, so now let's move on. We're moving on to sensory neurophysiology. So we're going to start to break things down into systems. And the system we're going to start with is the somatosensory system, our sense of our body, touch, pressure, pain, position, the sense of our body in the world. And then we'll move on to things like vision and hearing and balance and taste and smell. So we'll move on to the other sensory systems. So here's what we're going to do today. I'm going to introduce some general principles that's true for all of the sensory systems. So a global introduction to sensory neurophysiology. And here is a breakdown of some of the things that we'll talk about. And then I'll move into somatosensory, specifics of the somatosensory system. And here are some of the things we'll talk about. So here's an outline for today. And you can refer back to it when you review the project. By the way, the key concepts list for this section is, you may notice it's, 9 pages long or something. It's quite complete. So use that as a pretty thorough checklist of things to know for the next exam. So, like I said, we're going to do sensory neurophysiology. Today we're going to do touch, etc. Pressure, pain, proprioception, our body position, and then vision, hearing, olfactory system, and the taste physiology. And here are just some cartoons of, here's an image of the skin with some detectors for things like pressure or touch or vibration. Here is a cartoon of the somatosensory system that we'll break down later today or on Tuesday. Taste bud, auditory transduction machinery in the inner ear. Okay, like I said, general principles. Before we get into any specific sensory system, let's talk about some general principles and get some language out in the room. So we talk about 2 different types of sensory receptor cells. There are the so-called long receptor cells and the short receptor cells. So both of them have mechanisms for transducing some sort of sensory input into electrical signals that go to the brain, right? You know that when you smell something, the smell doesn't go to your brain. The smell goes to your nose, and then electrical signals go to your brain. So we transduce sensory input into electrical signals. action potentials. Action potential frequency is what we're going to transduce to. Long receptor cells, so the receptors that detect touch and other somatosensory modalities, pressure, pain, and the cells that detect smell, chemicals that get into your nose, are long receptor cells. They have, part of the cell has the receptor mechanism, and they have axons. That's the point here. Those cells have receptors and axons that transmit action potentials to the brain, and that's what makes them long. The short receptor cells, such as the photoreceptors in the retina, or the hair cells in the inner ear, or the taste cells in taste buds, the cells that have the receptors do not have the axon. The cells that have the receptors are short. They have receptors, they have neurotransmitters that are released, but then that neurotransmission to receptors, to neurotransmitter receptors on the afferent neuron trigger the action potentials. So in the long cells, the cells that detect the sensory modality are afferent neurons. In the short receptor cells, the cells that detect the sensory modality do not have axons, are not afferent neurons. They stimulate, or in some cases inhibit, the afferent neurons that bring the information to the brain. So I've used some of the language, long versus short, afferent. Afferent means it's going to the brain. When we get to the motor system, we'll talk about efferent neurons that take information from the brain to the body. Modalities is a fancy name for the different sensory stimuli. So let's talk about a long receptor cell. And so here is the stimulus. And in this case, we're either talking about a touch or pressure or pain stimulus for a somatosensory input, or a chemical that we can smell in an olfactory input, some stimulus. And that stimulus activates receptors that have evolved to detect that stimulus, whether we're talking about touch or pressure or a chemical. A receptor has evolved to detect that stimulus. And like neurotransmitter receptors, these create graded potentials, usually depolarizations. So the stronger the stimulus, the stronger the depolarization. A graded depolarization. So if we were to record, well, here it is. The stronger the stimulus, if we record at this point, sort of before the axon, actually this looks like it would be the axon hillock, which is where action potentials start. So just move in your mind, move this point up to where the plus sign is in the receptive terminal. So a small stimulus generates a small depolarization, and a large stimulus generates a large depolarization, graded, like I said. But it's a long receptor cell, so it has an axon, so it has the mechanisms for generating and propagating action potentials, and they are usually myelinated, so it happens fast, so the propagation is fast. And so if the receptor stimulus is below threshold, we get few or no action potentials. If it's below threshold, we'll get no action potentials. And a larger stimulus will give a higher frequency. If it's above threshold, we'll give a higher frequency of action potentials. And a higher stimulus will stay above threshold longer and give a higher frequency of action potentials. More depolarization, more frequency of action potentials. I think I asked you a question about this on the exam, sort of asking you to make sense of this even before I taught it to you. So here is, zero sum high frequency of action potentials coding the stimulus intensity. undetectable, low detectable, high detectable levels of stimulus. The receptor potentials code that information in amplitude, but the axon doesn't code that information in amplitude because it is an all or none thing, action potentials. It codes the strength of the stimulus in the frequency. And then as this propagates, meters in some cases, or, millimeters in the case of the nose, we don't see the baseline anymore. We simply see the frequency of action potentials. And so the information is encoded in action potential frequency. And no action potentials stimulate no neurotransmitter release at the end of that neuron. Low frequency of action potentials, a low amount of neurotransmitter release at the end of that cell, that afferent neuron. And high frequency, high release of neurotransmitter at the end of that afferent neuron. And then of course there's more processing, right? It's not, at the level that we're going to be talking about, we're going to be talking about simple inputs and outputs. You smell something, you detect it. But of course there's all kinds of cognitive events that happen too. You smell rose, you start to think about grandma or something, right? We're not going to get into that. Okay, let's do a little quick. Tricking most. I'll give you a clue. There are 3 correct answers. Nerve cells don't make myelin. Schwann cells and oligodendrocytes make myelin. Remember, the myelin comes from the cells that wrap the neurons. They have myelin on them, but they don't express. They have myelin on them, but those cells do not express the genes for myelin because those are coming from the glial cells that are wrapping them, right? So I tricked you. But now you know. Remember, I'll be honest with you. This concept is something that we tend to ignore, and then for those of you who, there have been many events where students have forgotten that glial cells exist, and they're about half of the cells in our brain, right? So they do important things like myelination. So I'm trying to emphasize, oh yeah, remember that other class of cells, the oligodendrocytes and the astrocytes and the Schwann cells. They do important things, including myelination. They express myelin, not the nerve cells. Okay, let's do this. Let's do the same question on this one. It's the same question, it's just looking at short receptor cells. Some, but not all of you in this one. And I'll give you a hint. There's 2 correct answers on this one. The short cells do not fire action potentials. So I didn't teach this to you, but there are no action potentials in the short cells. The receptor-dependent depolarization is transmitted directly. That depolarization opens voltage-gated calcium channels that cause neurotransmitter release, but there's no action potential in this cell. The first action potential is in the afferent neuron. Afferent neuron, action potentials, voltage-gated sodium channels. Afferent neuron, voltage-gated sodium channels. Short receptor cell, calcium channels, yes. Voltage-gated sodium channels, no. Yes, Kristen?
So would it be, as in,
Oh, they have a resting membrane potential, so
you don't need a voltage.
they don't need them, whether they express them or not, I don't know. They don't need them but they might have them anyway. I wanna be honest, that wasn't part of the question, was it?
No, it is not.
You're branching out, yeah. So, I wouldn't ask you about voltage-gated potassium channels, because I have a feeling it depends. Yeah,
So
sorry.
if they have T-stars and B-stars, does that mean that they release the neurotransmitter the same way?
Yeah, they do. They release the neurotransmitter the same way. The depolarization that is causing the neurotransmitter release from the short receptor cell depends on depolarization, and that depolarization comes from the sensory receptors themselves, not from an action potential invading a presynaptic terminal. The presynaptic terminal, it's short. The presynaptic terminal and the receptor end are so short that you don't need an action potential to get information from one end to the other. You can rely simply on depolarization and diffusion to get the depolarization from the receptor end.
So the receptor is what's opening those voltage-gated calcium channels?
The depolarization due to the receptor is what's opening enough voltage-gated calcium channels to cause neurotransmitter release at this synapse. Shahid, did that answer your question? Okay. All right, so let's talk about another concept that we will be using. This is a general concept, and that is the concept of a receptive field. So this is a cartoon of the somatosensory system, because that's where we're going. But this concept is true for other sensory modalities as well. So sensory neurons innervate a specific receptive field. And this is easy to imagine in the sensory system, where I can imagine that there are sensory neurons that innervate my palm, and I'm activating them now by touching my palm. And there are other sensory neurons that innervate my arm that I'm activating now, but I'm not activating the palm ones, right? So the receptive field of the neurons is determined by the physical location of where they innervate. And there are overlapping ones, right? So if I stimulate this part of my palm, I could be activating more than just one sensory neuron, right? But some of them are going to be highly activated, and some of them are only going to be partially activated because of the receptive fields overlap, but not, there's not universal overlap, there's just partial overlap. And so neurons that innervate the center of the receptive field are more activated than the neurons that innervate the periphery of the receptive field. Okay, so that's using some of the language. Receptive field. Sometimes a receptive field can be physical, like touch. It can be, so in the visual system, for example, I'm putting my eyeballs, my retinas are focusing on Eric's face. I'm looking directly at Eric's face. But I have a peripheral receptive field here. I can see the rest of the room, but not with not the same accuracy as I can see Eric's face, because I'm putting the center of my receptive field on Eric's face, but I'm seeing the rest of you fuzzy. Receptive field. That's the visual system receptive field. The olfactory system has a receptive field too. It's just not physical. I can detect the difference between this rose and that rose. Two different roses, two different types of, two different breeds of roses will have slightly different smells. So one of them will activate this olfactory receptive field more than another one will. And that allows me to discriminate between this rose and that rose, or between an orange and a tangerine. I can smell the difference between an orange and a tangerine. I think I can. Maybe we'll test it. Or between an orange and a lemon. You know, I can detect the difference. We can all detect. We can all agree that we can detect the difference between an orange and a lemon. There are some things that are similar. They both smell acidic, citrusy, but they're different. So they have overlapping, but not identical, receptive fields. Receptive fields can vary in physical size. So for example, again, in the somatosensory system, there are some areas of your body that have a very large receptive field, like on your back. I can touch this part of my back or this part of my back, and it feels exactly the same, because the receptive field on my back is very large. I can't discriminate. I can't tell whether I have two fingers. I mean, I can tell because I know the position of my fingers, but I can't tell whether I have two fingers touching my back or one finger touching my back when they're that far apart on my back. But obviously, on my fingertip or on my lips, I can tell the difference between two positions. So on my lips, I have a very, very small receptive field that allows me to determine the difference in position with high accuracy. And on my back, I have a very large receptive field that allows me to detect touch, but not with any high acuity or accuracy. So, high receptive field, small receptive field, high accuracy on my lips, large receptive field, low discriminatory ability on the skin of my back. And maybe we'll play around with this in lab. We can do some receptive field tests. Okay, here is an important concept in neurobiology that we're going to introduce in the setting of sensory neurophysiology. And that is the concept of lateral inhibition. Okay, so this thing called lateral inhibition sharpens receptive fields, allows us to make makes receptive fields smaller and sharper. Why would we want to do that? Because of accuracy, acuity. We want our fingertips to be able to determine a 1 millimeter difference between 1 and 2, whereas we don't care if our back tells the difference between a 1 millimeter, 1 or 2. We care whether it, I don't know, 10 centimeter difference. It's not that bad. So here's how this works. So let me walk through the mechanism of lateral inhibition. Let's talk about 3 afferent neurons. The one in the center is stimulated the most because it's in the center of the receptive field, and the ones on the side are stimulated less because they're out on the periphery of the receptive field. So high frequency of action potentials in the center one, and low frequency of action potentials in the peripheral afferent neurons. Those are the green ones. The red ones are illustrating the inhibitory interneurons that cause lateral inhibition. So the red inhibitory interneurons cause this phenomenon of lateral inhibition. So let's go through this. So and all the neurons are connected to their neighbors by inhibitory interneurons. So this afferent neuron is connected to this afferent neuron by an inhibitory interneuron. And this afferent neuron is also connected to this afferent neuron by an inhibitory interneuron. And this afferent neuron is connected to this afferent neuron by an inhibitory interneuron. And likewise over here, this one is also. Okay? So all the cells are connected to all their neighbors, not to all of their cells in the brain or anything, but to their neighbors by an inhibitory interneuron. Okay, high frequency here. High frequency here. Lots of neurotransmitter here. Lots of excitatory neurotransmitter here. Lots of action potentials here. Lots of inhibitory neurotransmitter here. This is an inhibitory interneuron, which means it inhibits, it has an inhibitory neurotransmitter, GABA, let's say. So this might be releasing glutamate, and this is releasing GABA. So a high frequency here is not only causing high frequency to the second order neuron, but it's causing high frequency in the inhibitory interneurons. These inhibitory interneurons are inhibiting this synapse. So this small amount of action potentials coming from the periphery of the receptive field is attenuated, going from 4 to 1, it's attenuated by this lateral inhibition. This peripheral stimulus is being attenuated by the lateral inhibition. And so now the throughput, the signal to noise is improved, is enhanced. We go from, I don't know, 10 to 4 to 10 to 1.
There's still 10 because you have inhibition from the right and left one too, don't you?
Okay, so it's not quite 10. So that, let's say this is 12 to 4, and this is 10 to 1, because this, you're right, this small amount of stimulus in the periphery is causing a small amount of inhibition here. And so this inhibition is less than this inhibition, but it's not 0. And so this inhibition is reducing this throughput a little bit. But this stimulus is inhibiting this throughput a lot. And that's what causes an enhancement of the a sharpening of the receptive field edges. Aiden, did you have a question?
Yeah, I know you mentioned noise and signal. Which one are you considering to be the signal here?
The signal is the center of the receptive field, and the noise is in the peripheral part of the receptive. We're trying to sharpen the receptive field. So the edge is the noise, and the center is the signal.
Okay, fair enough.
So I hope I've made the concept of lateral inhibition plausible and understandable in this simple somatosensory system. We're going to see it in the retina when we talk about sharpening visual receptive fields. We're going to see it in the auditory system when we talk about sharpening auditory receptive fields, our ability to detect, you know, an A440 tone from a flat A440 tone. That just sounds bad because it's flat. Those of you with perfect pitch know what I'm talking about even without me playing a tone.
I guess I was curious as to like how the central axon, that being like a little bit kind of inhibited, like how inhibited and why that's like advantageous. Like,
Oh, so you mean why is it advantageous to have this
Yeah.
inhibition? Well, because sometimes this is the center of the receptive field, right?
Oh, yeah.
Sometimes this is the center and this is the periphery, right? We don't, there's no, this is just an example where this is the center and this is the periphery. But after all, that's an example. It could be, you know, instead of touching, I mean, I want to be able to detect this touch and this touch and this touch. So this is the center and this is the surround. But sometimes this is the center and this is the surround. Okay? So we're going to come back to this repeatedly, but this is foundational. Make sure you understand it. So you already know from studying neuroanatomy, the brain, that we have different parts of the brain that are dedicated to different sensory modalities, different sensory inputs. So for example, the primary visual cortex is in the very back of the occipital lobe. The primary auditory cortex is in the superior temporal lobe. It's in the temporal lobe. Some of you wanted to get credit for saying this is the auditory cortex. And I didn't give you very many points for that because it's in this superior gyrus of the temporal lobe. The somatosensory cortex, which is where we're going now, is in the post-central gyrus. We'll talk a little bit about the taste cortex. We'll talk a little bit about the olfactory cortex, which is not on the slide because it's embedded, it's inside. So it will be useful to know that the auditory cortex, the primary auditory cortex is in the superior temporal gyrus. The visual cortex is in the most posterior part of the occipital lobe. And the somatosensory cortex is in the most anterior part of the parietal lobe, which is the post-central gyrus. Anterior parietal, post-central. Okay, so now we're getting into somatosensory systems. We're transitioning away from general and into specifics. We have 5 somatosensory modalities. We can detect fine touch, light touch, pressure, pain, temperature. I forgot to mention that so far, but you know you can detect hot and cold. And body position, the position of your body in space. You know where your body is, even if you're not looking at it. These 5 sensory modalities are lumped into 3 pathways. So we don't have 5 different pathways, we have 3 different pathways, but they are different. The touch, pressure, and vibration pathways, the touch and pressure, which also includes vibration, is one pathway. And we'll talk about the anatomy of that pathway soon. Pain and temperature, and also itch, but pain and temperature are lumped together into another pathway. And proprioception, body position, proprioception is the name that I will use for that. Write that out. Proprioception is the fancy name for knowing where your body is carried in yet a 3rd neural pathway. So 3 pathways, so you do want to know that touch and pressure go together, that pain and temperature go together, and that body position is separate from those 2. We have what are called dermatomes, so derma, skin, dermatomes. Tome just means slice, so skin slices. Different parts of this body are sliced, whatever, have different dermatomes. So for example, you know that there are cervical and thoracic and lumbar and sacral vertebrae that have, and those are the sections of the spinal cord as well. Touch receptors from, let's say, the shoulder and the upper chest. Those neurons, those afferent neurons, enter the spinal cord at C4. And that defines a dermatome. The skin around your belly button, those afferent neurons enter at thoracic 10. And there's a slice. And it goes around the back too. It's a ring of skin that is innervated by afferent neurons that project to the T10 level of the spinal cord. Now, will I ask you to know which one is which? Will I ever put up a blank one of these and say, what is that dermatome? No, I won't do that. But I might ask you to apply this image to solving a problem related to where the information is entering the nervous system. entering the CNS, the central nervous system. Okay? So I do not want you to start to memorize dermatomes, but I do want you to understand what I mean when I use the word dermatome, and to understand that information from the thumb enters the body, enters the central nervous system at a different point than information from the forearm, from the arm. Information from the thumb enters at C6, and information from the pinky enters at C8. So if there was a damage in the spinal cord, you might have differential sensation abnormalities in the hand.
How specific should we know that? Should we know like the core is thoracic and legs are?
No, you don't have to even know that. If I asked you to work with this, you would have this image or something like it to work with. I guess I'm giving you a heads up. There is a problem in the next problem set that will ask you to use this image, but you'll have it in your PowerPoint. So C1, I don't even know where C1 is. V1, 2, and 3. This is the, V is not a V, it's 5. This is the trigeminal nerve. Okay, dermatomes. So now let's talk a little about the receptors in your skin that detect touch, pressure, and vibration. So here's a cartoon from your textbook. Here's the epidermis. Here's the dermis of the skin. There are 4 types of these so-called corpuscles that detect pressure, touch, or vibration. Meissner's, Merkel's, Pacinian, and Ruffini. They detect slightly different modalities. Touch and pressure, vibration and deep pressure, stretch. I don't want you to know which is which. I do want you to recognize these names and maybe even be able to report in a quiz the names of some of them. So for example, if I might say, there are 4 corpuscles in the skin that detect touch, pressure, vibration, or stretch. Give me 2 of them. So remembering Meissner, Merkel, Pacinian, Ruffini could come in handy for a couple of points. The main concept is that there are these different corpuscles. Are these short or long neurons? Short or long receptors? They're all long. So this is the receptive end, and here's the axon, the myelinated axon that is going to the spinal cord. D is the deep one, right? Pacinian corpuscle. A is a shallow one, touch and pressure. E is the vibration one, right? So this is obviously cartoon. And if I were to show you a micrograph of a section through the skin, it would be really hard to see. They're hard to see. But these are the receptor cells. These are the receptor ends. And here's the myelinated axon that carries information about touch and pressure from the skin to the central nervous system. Now you can imagine, okay, how does your body detect something as weird as touch? There's no ligand for touch. The bottom line is that there are things called stretch-activated channels. So when I squeeze this, I change the stretch of the membrane. And some of these have multiple membranes. And so if I squeeze this, I'm changing the stretch of the cell membrane. I'm activating stretch-activated channels, which then, depending on whether it's a stretch-activated sodium channel or a stretch-activated potassium channel, changes the action potential frequency in the afferent neuron. So there are stretch-activated channels, and that's key to understanding the molecular mechanisms for transducing touch into action potentials, or pressure into action potentials, or even vibration, sort of texture of a field. Right? When I run my hand over sandpaper, it feels different than when I run my hand over a mirror because of the difference in vibration that causes in my skin. In this cartoon, they're called free nerve endings. So the so-called free nerve endings are what transduce pain and temperature. And there are receptors in the free nerve endings that detect pain and temperature. So, for example, in free nerve endings that detect heat and also painful stimulus, there's a receptor called TRPV1. It's the capsaicin receptor. So some of you know that hot chili peppers produce a chemical called capsaicin. Capsaicin is the chemical that activates the capsaicin receptor, the TRPV1 receptor, and gives us the sensation of heat. You may know that eating temperature hot chili feels painful, whereas maybe eating cold, I'm talking about a really hot chili, eating a cold chili might be less painful, because these receptors detect both heat and capsaicin. And it's because it's a graded response, it's additive. So if I take a hot chili pepper and it'll be hotter, it'll feel hotter than a cold, an ice cold hot pepper. Right? Yeah,
Are you talking about hot as spicy, or are you talking about hot as heat?
both. So I will use the word spicy from now on, and I will use hot for temperature. So a hot, spicy pepper will cause more of an activation than a cold, spicy pepper. And some of you who have grown up eating spicy food may know that when you go to Gulzar's, you order a 9, and when your friend who didn't grow up eating spicy food goes to Gulzar's, they say, I can't eat that, 2, please, for me. And you can't even share because one tastes like nothing and the other, for one person, and the other tastes like burning fire to the other person. And that's a result of lifelong changes in your sensory system. Those of you who have grown up eating hot, that is to say spicy foods, have in fact killed off a lot of your sensory neurons in your mouth. And that's because of the action of the TRPV1 class of capsaicin receptors. So it's a receptor both for the chemical and for heat. It detects heat. There was an interesting story about a scientist who was the scientist wanted to image MRI, do an MRI imaging study of people in pain. But you're not allowed you're allowed to hurt people, but you're not allowed to cause irreversible damage when you hurt people in an experimental setting. So what this experimenter did was apply a capsaicin cream, which felt moderately uncomfortable, but wasn't extremely painful. And then to evoke extreme pain, put a warm compress on that area. That combination caused intense pain, and then that could be imaged. But it was reversible because the capsaicin cream by itself didn't kill the neurons, and the hot compress was just a warm compress. So it was extremely painful, but it wasn't damaging. At least not for the short time that the study was done. So the point is that that's an example of the additive nature of the heat and the capsaicin activation of capsaicin receptor, of TRPV1. And we have other receptors that detect painful stimuli. We have another receptor that detects tissue damage and inflammation. We have another channel that detects damage and inflammation. Ischemia means low blood flow, which is painful. If you put a tourniquet on, it hurts. And we have other receptors that detect cold temperatures. And they also detect a chemical like menthol. So, for example, icy hot, that compress that you put on if you've had a muscle pain, has both capsaicin receptors has both capsaicin and menthol in it to try to sort of desensitize all of the sensitivity of that part of the skin, trying to decrease the sensation of pain by sort of overwhelming it with hot and cold.
So is that one helping too?
Yeah, right. So there's an interesting story, which might be apocryphal, that the Coca-Cola company was trying to develop a chemical that activated TRPM8, but did not taste like menthol. You can see why. If you could add this chemical to Coke, it would taste cold, even if it was at room temperature. It failed. They could not find a chemical that activated this receptor that did not trigger this taste response. And obviously you don't want your Coke to taste like menthol. Yes?
Regarding just the acid sensing ion channel?
Yeah,
So when you have no blood flow, is there like an accumulation of acid?
exactly.
Okay.
Ischemia triggers an accumulation of acid, which activates this channel, which is why it hurts. And then we have all kinds of things that modulate, augment this painful response. So what do I want you to know? I do want you to know that TRPV1, the capsaicin receptor, is the hot detector. And I think I would give you everything else, in a question. I would say, remember, TRPM8 is the cold receptor and TRPV1 is the hot receptor, and then ask you to solve some problem with that information, just like I did on this exam. Okay, let's do another clicker, and then maybe we'll take a break. Not maybe, we will take. Let's do a clicker and take a break. So I'm introducing a concept here, the name of the neurons. Well, there's a class of neurons called nociceptors. That's what I'm introducing here. Aiden?
Is there multiple answers for this?
Oh, that's usually true. I'll give you a hint, 2 on this one.
Oh, come on, there's 3.
What?
Shouldn't there be 3 at least?
No, not on this one.
No,
Touch and pressure do not activate pain receptors.
Wait, really? The pressure would really hurt
If you hurt so much that, if you're pressing so much that it's hurting, you're causing tissue damage, and that's activating the nociceptors, not the pressure sensors.
Oh.
Okay, which of the following proteins is likely to be expressed in Meissner and Merkel corpuscles? I think I gave this. This one's pretty, there's one correct answer to this one. Well, yeah. Remember that the thing that allows those corpuscles to detect pressure and stretch and things is that there are things called stretch-activated ion channels. Okay, let's take a break, a couple minutes, and we'll see, then we'll switch over to proprioception. Perception. The ability to detect your body position, even without looking at it. The proprioceptors, the cells that transduce proprioception, are in your muscle. In fact, they are specialized muscle cells. So here's a muscle, tendon, bone. Embedded in the muscle cell is a specialized muscle cell called the muscle spindle. And then there are afferent nerves that wrap around the muscle spindle and provide information to the brain, to the central nervous system, about the stretch and length of that muscle. So your muscles, speaking globally, your muscles are not only things that cause you to move, but they have embedded in them the sensors that allow your brain to know where your muscles are in space. And then you have learned, okay, if I get this kind of signal, my elbow is partially flexed. If I get this kind of signal, whatever it is, I can't tell you what it is, but because I've learned that my elbow is extended. And babies learn this, right? Babies, they're not born knowing how to move their body. They figure it out. They learn that, oh, if I do this, I can bring my hand to my mouth and eat the Cheerio. So, bottom line here is that muscle fibers, which we will talk about more when we get to the motor system, also contain muscle spindles, which are essential for proprioception. And we have afferent neurons that carry information from the muscle to the brain, reporting information about the position and the stretch of that muscle. And we have efferent neurons, motor neurons, that innervate the muscle and cause the muscle to contract. So when the muscle contracts, the stretch of the spindle changes, and that information of stretch change is transmitted to the brain, because the spindle is embedded in the muscle. And the spindle can also shorten and lengthen so that it stays in sympathy and in synchrony with the muscle around it. But its job is not to cause movement. Its job is to report movement back to the brain. We'll talk more about muscles when we get to the motor system, but we're talking now about the sensory system, the muscle spindles embedded in the muscle. Any questions about proprioception? Do you think they have stretch-activated ion channels? Pretty good bet. Because the muscle spindles are detecting the stretch and position of the muscle surrounding. Okay, so let's talk some more about those neural pathways that carry sensory, somatosensory information. So we're talking about somatosensation. Remembering that there are different modalities. Touch, pressure, vibration is one. Well, touch, pressure, vibration are three modalities, but they are lumped together into one pathway. Pain, temperature, and itch are three different modalities. When you have an itch, it's not the same as feeling pain or temperature, but itch, pain, and temperature are carried in another neural pathway, and proprioception is carried in yet another neural pathway. Okay, so let's get oriented. You're going to see images like this a lot, and they can be confusing, so I want to get oriented. Here is a horizontal section of the spinal cord. And you'll see on the last page of your handout a blow-up version of this horizontal section of the spinal cord. Gray matter is in the central area, this butterfly-shaped thing in the middle is gray matter. This is where the cell bodies and dendrites are. This is where the synapses are. And white matter is in the periphery of the spinal cord, and that's where the myelinated axons are. So this is a horizontal, this is sort of an oblique view of a horizontal section. Your handout has just a flat horizontal section. Horizontal section. This is a coronal section of the brain. So we've changed orientations completely. Okay? Horizontal section, coronal section. The scale is wrong, obviously. I mean, this scale is different from this scale. Okay? But we'll use this cartoon frequently. So now let's talk about the pathway that carries both pain and temperature and itch information from the periphery, from the skin, pain, temperature, and itch from the skin to the brain. Let's just walk through this. So this is the afferent neuron. Remember, it's a long neuron. It has a receptive field in the skin, and it has an axon that's going to enter the spinal cord. Its cell body, and I want you to label this image on your handout. It's the cell bodies of these afferent neurons is in this ganglion called the dorsal root ganglion, DRG. That's where the cell bodies of somatosensory afferent neurons are. So an axon, these are bipolar axons. You learned this name, I think, way back at the beginning of the course. So they have a receptive field, a receptive terminal. They have an axon. They have a cell body, sort of in the middle of the axon. They have more axon on the other side of the cell body, and then they have their presynaptic terminal here in the spinal cord. So sensory receptor out here, not shown, but you can illustrate that on your handout. Axon, cell body in the DRG, axon, presynaptic terminal. Let's say that little V is. Presynaptic terminal. The first synapse of the pain, temperature, and itch pathway is in this part of the spinal cord called the dorsal horn. So I do want you to annotate that somewhere. This area right here is the dorsal. So here is the right dorsal horn, and here is the left dorsal horn. Here is the, I'm doing it over here. Here is the right dorsal horn, and here is the left dorsal horn of the spinal cord. Gray matter. Dorsal, ventral, dorsal horn. So let's get oriented. Dorsal, ventral, left, right. You're looking at my spinal cord. So that's why this is the right, and this is the left. This is the back, dorsal. This is the front, ventral. This is the dorsal horn.
So that's where the synapses are?
This is the first synapse of the pain and temperature pathway is in the dorsal horn. The second order synapse, the next synapse, so we got to get this pain to our brain somehow. Otherwise it doesn't do us any good. And pain, let me just say, pain is good, right? Because you want to be able to detect pain. If you put your hand on a hot stove, you want to get your hand off that hot stove as fast as you can. Because you don't want to cause more pain, more burn, more tissue damage than necessary. People who are born without pain pathways don't live very long. Because nothing hurts. They jump off of buildings. Because they say, oh, I can do that. You know, imagine a 10-year-old. I can jump off this building and it won't hurt. Well, it won't hurt, but you'll die anyway. Anyway, pain, that's why pain is adaptive. Now, chronic pain is maladaptive, and we probably won't get into that. But pain is good for us because it prevents us from doing things that we shouldn't do. If we've broken our arm, we're going to take, even before we go to the hospital and get it set, we're going to take action to try to minimize the pain of the broken arm, which will minimize the damage caused by flow. Maddie?
DRG stood for what again? What was the, like, full thing that DRG stands for?
Dorsal root ganglion. Dorsal, because it's on the dorsal side of the spinal cord. Root, because ganglion, because that's what it is, a cluster of nerve cell bodies. DRG. Okay, so the first synapse is in the dorsal horn. The second order neuron crosses over to the contralateral side. So this is a crossing over here. Contralateral white matter in an area of the spinal cord called the anterolateral column. Anterolateral spinal cord is where the axons, the myelinated axons that go up the spinal cord to the brain are. Pain and temperature information is carried in the spinal cord in the anterolateral column. Antero, towards the front, lateral. Contralateral. It's going to make synapses in the midbrain. It's going to make synapses in the thalamus. The thalamus is going to process this painful information, and this is the third, now it's the second synapse. We're activating the third neuron that carries information from the thalamus to the somatosensory cortex in the post-central gyrus on the contralateral side. So pain information on the right side is processed in the thalamus and the somatosensory cortex on the left side. Left is contralateral to right. I do want you to know where the first synapse is, where the crossing over is, and where they go.
What do you call the crossing over?
The crossing over is in the spinal cord. It's got a fancy name. Let's leave the fancy name aside. The second order neuron crosses over in the spinal cord. Let's talk about another main pathway, touch pressure and some of the position. Touch pressure. Afferent neuron, dorsal root ganglion, axon, no synapse. It goes up so this neuron, this axon does not synapse, it goes up the ipsilateral dorsal column. This is white matter. If you look on the other image, you'll see that this area here is the dorsal column. An area of information on touch and pressure from the right side goes up the right ipsilateral dorsal column. It makes a synapse in the brainstem, and that's where crossing over happens. Crosses over now this is the first, this is the second neuron, so the second neuron crosses over, just like the second neuron here crosses over, but here the second neuron crosses over in the spinal cord, and here the second neuron doesn't even start until the brainstem and crosses over in the brainstem. Crossing over in the brainstem, thalamus, somatosensory cortex on the contralateral side, because we have crossed over. But the location of the crossing over, here the crossing over was in the spinal cord, where the information entered. This could be at L5 if the information is from the leg, and this isn't until we get into the brainstem. Right? So if I have a painful stimulus in my right leg, it's entering the spinal cord at let's say L5, in the lumbar. And then it's going from my lumbar spinal cord to my brain on the contralateral side. If I have a touch pressure information from my right leg, it's going to my brain on the right dorsal column in the spinal cord. and it doesn't cross over until we're in my head. So I do want you to know the information that's embedded in this slide. We will talk about, there's going to be another pathway for position called the spinocerebellar tract. I don't think I think we're going to not do that today. So here's an image from your textbook. Let's just get oriented. This is a horizontal section. It shows the spinal cord embedded in the vertebra. Here, what have I called this thing? So this, here it is, dorsal root ganglion. Here's the dorsal root, I guess, okay. Dorsal root ganglion, dorsal root. Here's the dorsal horn. So this is a slide that illustrates the language that I've been using. Here is a cartoon of a section of the spinal cord. And you will use this cartoon in the next problem set. So let's just spend the last couple of minutes getting oriented to this cartoon. Dorsal column. Where do I go for the dorsal column? Well, let's start with I label. This is right, left, dorsal, ventral. Dorsal horn, dorsal horn. What do you suppose we're going to call this when we get to it? The ventral horn. Dorsal column. This is the dorsal column. This whole thing is the dorsal column. This is the right, and this is the left dorsal column. Touch pressure information is in axons going to my brain on the ipsilateral dorsal column. Anterolateral, which also has the name spinothalamic. Anterolateral. So dorsal, dorsal, lateral, antero. Anterolateral is in there somewhere. Left, right, anterolateral. So if I were to cartoon pain information from my right side, pain information, dorsal root ganglion, enters synapses. Remember we're talking about pain, synapses. The second order neuron crosses over, and then its axon goes to the brain on the contralateral, anterolateral column. And we'll talk about spinocerebellar later. Oh, did I give you this one? This one gives you all the information you'll ever need to know about the organization of the spinal cord. So you can use this as a reference. So here's the dorsal column, but this has more information. Now, let me emphasize, this has more information than you need to know. So for example, the dorsal column is organized into sacral, lumbar, thoracic, cervical. You don't have to know that organization. The anterolateral, so there's anterolateral. It also is organized into sacral, lumbar, thoracic, cervical. You don't have to know that level of information. But you do have to know generally where the anterolateral is. I said, when I drew it before, I said this block right here. That was only partially correct because anterolateral is sort of in this area of the white matter. Okay, let's stop. Yeah, let's stop. That's the end of the slideshow. This, of course, as always, this will be posted to Moodle. And your grades have already been posted to Moodle. And I can give you back your quizzes and your exams.